Hi, everyone. I am Kelly Hagman, one of the co-chairs of the Genomic Technology SIG. Welcome to the Genomic Technology SIG quarterly webinar series. Today we're talking about bioinformatics. We're going to get started in a couple of minutes. Just going to wait for the top of the hour and for a few more attendees to hop on. Okay, hello everybody, and again, welcome to the Genomic Technology SIG quarterly webinar series. We're going to be talking today about bioinfo what, or bioinformatics. We have a couple of speakers today that we're really excited about. So we have Dr. Bryce Sarver and Carla Brigatti. I'll be introducing them before each of their sections. Again, I'm Kelly Hagman. I'm one of the co-chairs of the Genomic Technology SIG, and I'll be your moderator today. I'm going to go through a couple of logistics before we get started. So today's webinar has been approved by the National Society of Genetic Counselor for 0.1 CEUs, or 1 contact hour of Category 1 CEUs. A few logistics I'd like to go through is that you'll see the control panel that appears on the right of your screen. It looks like this. From the audio pane, you can switch from telephone and computer to computer, mic, and speakers, depending on how you'd like to listen. Most of you probably already have that figured out by now. And then over here, there's a section that says questions. Please ask all of your questions in this section. You can ask them throughout the talk at any time, and we'll gather them up, and we will have a Q&A at the end. So we will reserve all the questions for the end of today's talk. There's an evaluation that pops up in a web browser once you log out of the webinar. Make sure you fill that out in order to get your CEUs. I'd first like to introduce Dr. Sarver. He is a bioinformatics scientist at Ambry Genetics. After graduating from Washington University in St. Louis with a major in biology and philosophy, Dr. Sarver received his PhD in bioinformatics, evolutionary genomics, and phylogenetics from the University of Idaho. Afterwards, he completed a postdoc focused on computational genetics and mouse evolutionary genomics at the University of Montana. Dr. Sarver is interested in the development of techniques for the analysis of genomic data in non-model, near-model, and model systems, especially using high-performance computing environments and relational databases. In particular, he's curious about how new and existing computational techniques can be leveraged to identify the genetic basis of human disease and build genomic data sets in non-model systems, such as those lacking a reference genome. He's led or contributed to a number of studies in a variety of systems, including mice, chipmunks, bears, and fungi. All right, I'll pass it off to Dr. Sarver.
Cool, thanks, Kelly, for the invitation to present. Just to get started here, so for those of you who don't know me, I'm Bryce. And what I'm going to be covering today is a pretty general overview of a variety of concepts and the data structures associated with them that are actually used in the analysis of next generation sequencing data, particularly with respect to calling variants that can be used in either a clinical or non-clinical research framework. So just full disclosure, I'm a full-time salaried employee of Ambry Genetics, but nothing I'm talking about today is related to Ambry Genetics. So this is going to be the structure of the talk. Basically, I'm going to start off by kind of bridging the gap between the lab and the computer, that is, going from the molecular side of things into the actual analysis of bioinformatic data, which primarily takes place on the computer. Then I'm going to transition into developing an overview of pretty basic NGS pipeline for variant calling. And then as part of that pipeline, I'm going to describe the general steps and data structures that we're working with, starting with raw reads and then mapping those reads and then actually calling variants and then describing how that data might actually be distributed to end users. And then if we have time at the end, I'm going to wrap up with a couple additional considerations before handing it off. So when I started my PhD, I spent a lot of time in the lab doing Sanger sequencing, actually. And it was around kind of the late 2000s when we began to transition into high throughput sequencing technologies that most of the work couldn't actually be done on the benchtop and then be followed by a little bit of informatic analysis, like traditional Sanger analysis might take place. It actually needed to be primarily done on a computer in the first place. So there's kind of this paradigm shift, and what that means is that bioinformatics kind of requires this additional computational skill set that I see more and more students as they progress through graduate school currently are beginning to develop, and ends up being really useful for the analysis of this high throughput and kind of genomic sequencing data in a general sense. So before I get into some details, I want to make sure that we're on the same page just with a couple of general terms you may have heard or may not have heard before. The first is a library. So a library is basically DNA or RNA converted to DNA that has been prepared for sequencing through fragmentation and adding on a couple of additional oligonucleotide sequences. So you can imagine taking all of your genomic information, all of your nucleic acids, and you fragment that in the lab, and then you tack some additional stuff onto the end. This is generally what we refer to as a library. And when we take these fragments and we actually perform sequencing on them, then we're working with what we call reads. And reads are kind of the atoms of bioinformatics, like atoms actually are in chemistry. And a read is a single partially sequenced fragment. So each fragment can be sequenced either in one direction or in both directions. If it's sequenced in one direction, it's referred to as being single-end or single-end sequencing. And paired-end sequencing, if it's been sequenced in both directions. Paired-end information contains a little bit more general information about the fragment in its own right, because we know that those two reads are connected by virtue of being part of the same fragment. And finally, a reference is, very generally speaking, a collection of genomic information used for analysis. And a reference can be an entire genome, like the human reference genome. It could be an incomplete genome, so it doesn't have to have all sequences for all chromosomes or anything like that. It can be targeted molecules. A good example of this is the mitochondrial genome. Or it could be collections of other fragments. Maybe you're working with a reference transcriptome, or maybe you just have collections of basically chromosome-like fragments in other systems that aren't even closely related to humans and have no other genomic resources available with them. So many bioinformatic workflows generally start from the same place, and they're using very similar data structures. And the difference between the different types of workflows really depends on what type of analysis is being conducted. So if we're doing a DNA-seq analysis, which encompasses whole exome sequencing, whole genome sequencing, that type of stuff, we're really interested in calling variants. We're starting from these pools of sequenced reads, and then we're using statistics and other methodologies to go through and identify variation. But maybe we're interested in different levels of expression, so we're looking at an RNA-seq analysis. In that circumstance, we're not necessarily going to be calling variation, but we will be interested in different relative counts of the number of reads for a particular gene of interest. Maybe we're interested in how DNA binds to proteins and do a ChIP-seq analysis, protein structure for ATAC-seq. Maybe we're interested in methylations with bisulfite sequencing, that kind of stuff. But regardless of the technology we're using, we're really still starting with those atoms, those pools of reads in the first place. And the bioinformatician just needs to know, okay, what is this? What am I working with? And what technology should I be using in order to analyze the data at hand? So let's, from first principles, build up kind of a general NGS pipeline. So once the library is prepared, it's thrown on a sequencer like this Illumina HiSeq 3000. Some sequencing is performed, a bunch of chemistry takes place, and then at the end of the day, you get a raw binary data structure. That's called like the raw base call format. It's presented in a sense that makes, or in a format that makes sense to kind of the machine itself, and it's pretty efficient, but it doesn't really mean a lot to a human. So I'm going to ignore the BCL format, and we're going to start from actually the raw reads themselves. So the raw data can be converted to reads, and the reads are stored in what's called the FASTQ format. Then we take those reads and we place them in a genomic context. That is, we get the chromosome and position that each read, of the location where we think each read is housed in the reference genome. And the reads that come out of that are called mapped. The process is called mapping or aligning, and this is in the sequence alignment map format, or the SAM, or its binary representation, the BAM. And then once that's done, we can perform statistical variant calling and take a bunch of additional information into account. And at the end of the day, we get our raw variants, and that's the sort of the variant call format, or BCF. So what does a read actually look like? Well, a raw read generally consists of two connected parts, and that's the nucleotides themselves, so the actual ACTG bases, and then a measure of our confidence in the nucleotides. And in the FASTQ format, which is how these reads are sorted, it contains a lot of information, and each read takes up exactly 4 lines. The 4 lines are as follows. So there's a sequence identifier, which starts with the @ sign. Then there's the nucleotides. Then there's what's called the quality score identifier line, which is just a plus sign. And then there's the quality scores themselves, that measure of our confidence. So let's go through each one of these in turn. So the sequence identifier might look something like this, and this can change depending on chemistries and different platforms and that sort of stuff, but the general principle is the same. So it starts with an @ sign, and it contains some information about the instrument that it was run on, what run on that instrument it might have been. If we're using Illumina technologies, something about the substrate itself, the flow cell that it's used, which lane of the flow cell, where in that lane, etc. So a read sequence identifier really is a unique piece that provides a lot of information about the data that we're working with. Additionally, some other information can be tacked onto it as well, whether it's single or paired. Maybe in older technologies, it might have been filtered out or used as a control read or something like that. May even include the sample name in and of itself. So it's kind of a general description line of the data that we're working with. Next comes the nucleotide sequence. It's pretty straightforward. It's just all the letters. Next is that plus. It's pretty straightforward as well. And the last is the quality scores themselves. Now, quality scores, technically defined, are ASCII transformed Phred scale probabilities that a base call is incorrect. There's a ton of jargon in this statement, so let's go ahead and break it down. So quality scores are represented in a string of characters that, one example might actually be this, and each one of these symbols actually corresponds to each one of the nucleotides that's present above it in the file. And then we calculate what's referred to as the Phred scale probability that a base call is actually incorrect. If you remember high school algebra, we can calculate it using these equations. So probability that a base call is actually incorrect. So if we're 100% sure that a base call is wrong, the probability is going to be 1. And then log base 10 of what equals 1. That ends up being 0. Negative 10 times 0 is 0, so our quality score is 0. If we think about it from another side and say, maybe we think that there's only a 1 in 1,000 chance that our base call is incorrect. Now we're dealing with 10 to what power equals 1 over 1,000, and then that's minus 3, and at negative 10 times negative 3 equals a quality score of 30. So that's how you Phred scale the qualities. But then in order to actually get the representation, you add some value to it. So using Illumina 1.8 technology, we're adding 33 to it, and then converting it to an ASCII character. And if you're not familiar with character sets and that type of stuff, you don't need to worry about the details. All you need to know is that there's a correspondence between the number and the character that we see on our computer. An example of it might be like this. So if our quality score is 0 in the first row, take it and add 33 to it, you get 33, and that symbol is an exclamation point. And if it's 40, take it, add 33 to it, get 73, that symbol is an I. So that's a really efficient reduced representation way of including a measure of our confidence in concert with the genetic data itself. Just to show you what it looks like on a computer, most of this is done in Unix-like environments because these file sizes are very large, but this is what you might see. So you see the reads basically repeat every 4 lines, and everything's just kind of all the way down. This is a non-compressed format so that we can visualize it here, but often you'll see each one of these file types in their compressed representation just to keep file size down. Okay, so that's the raw data. Now we really want to get something out of this. So what we're going to do is attempt to take those raw data and place it in a genomic context. So for almost all analyses, really you want to take the read and you want to figure out where it exists in the reference genome with a high probability. So finding the optimal placement of where this actually is housed in a reference genome among 3 billion other letters, say in the human reference, is computationally challenging. So you can imagine trying to take 100 characters and figure out where it exists in among 3 billion other ones. That's a question for not only mathematics, but also computer science in general and algorithms and that sort of thing. So it's very computationally intensive to do so. And this is technically called mapping, or sometimes you see alignment, although alignment has some different technical meanings in other subfields of biology. And it requires a reference genome. I think that's kind of implicit in placing it in the genomic context in the first place. But there are a number of popular programs that do this. You might have heard of some of these before, BWA or Bowtie2 or Novaline. So just as a graphical example of what we actually want to do, imagine you got a reference genome here in blue, and then you have 8 reads here. These are just single-end reads. We will ignore paired-end reads for now. Imagine you're going to go through, and for each one of them, try to come up with our best guess of where everything aligns to the reference genome. So we're really interested in kind of chromosome or scaffold if you don't have true chromosomes, and position. But there are actually 3 cases that can take place here. So you can either take a read and place it in a genomic context uniquely. You could take a read and maybe it maps with equal probability to multiple locations, like read 7 here. So read 7 shows up twice. Or maybe the read is really low quality or just very divergent from your reference sequence, and maybe we can't place it at all. So the statistical techniques that we deal with variant calling will take some of this information into account. Now, all of this information is reported back at the end of the day in the sequence alignment map format, the SAM format. And this format includes all the genomic information plus all the information in a FASTQ file. And these files are pretty large, and so again, they're stored in their binary representation, the BAM. And this is what kind of the end-all data structure that when we're thinking about placing reads in genomic context or visualizing the reads themselves, most people talk about working with the BAMs because there is such a reduced file size with these. Now, BAMs contain 2 parts. First is a header section with information about the reference, program use, what kind of samples we're dealing with, that kind of stuff. And the second is an alignment section. And this is what really contains the meat and potatoes of the BAM, where in the genomic context each read is positioned, chromosome position, some quality, some measure of competence, that kind of stuff. So as an example, let's take a look at a header. This header is from 1000 Genomes Project. The header fields basically all start with an @ sign. And the first one is basically this header line, and it contains something about which format we're dealing with and the sort order of the various positions of the read in the file itself. In this example, we're also including something about the reference sequence dictionary. So basically, this is the chromosomes, how big they are, that kind of stuff. And then a little bit of information here at the end about read groups. Read groups are basically sample information, can include stuff about sequencing platform, that kind of thing. The alignment section is a bit more complicated, and it can actually differ from application to application. So basically, there are a number of fields that are required, and there may be more. But the really important required ones are the read sequence identifier, which is just the name of the read, the same name from the FASTQ. A bitwise flag, which tells programmers a little bit something about the record. The chromosome reference, the chromosome or reference sequence, and the position on that chromosome. So for example, chromosome 1, position 100 million. And then just like we had a measure of confidence for our base calls, we also have a measure of confidence in our mapping. And this is referred to as mapping quality. Now, interpreting mapping quality can actually be a little bit more tricky than it is for interpreting other types of qualities because it's calculated differently depending on which programs are actually used to do the mapping. And importantly, it might not even be a measure of confidence about mapping. It might just be, for example, if you're using splice-aware aligners for expression type studies, it might just be the number of counts or the number of times that this read is placed in the reference genome in a very general sense. So technically speaking, from the specification, it equals this Phred scale probability that the mapping position is wrong, rounded to the nearest integer. And a value of 255 indicates that the mapping quality is not available. This could be it doesn't map, or reads can also have their mapping quality shifted if they map multiple times, for example, depending on which application is being used. So when it's actually reported, it's likely a function of a bunch of different things, including the reference sequence. Is that region hard to align to? Base qualities, details of the algorithm, that sort of stuff. And then the last remaining fields include things like that are a little technical. I won't go into too much depth, but there's a way of representing how many matches and mismatches take place for a particular read. If you're doing paired end sequencing or other types of studies, the name, position, and basically length that we're dealing with for the next reads in the file. Then there is the nucleotides themselves and the quality scores themselves. So if you remember, this basically means that we're including everything in the BAM file that was in the FASTQ file. So you can go from BAM to FASTQ pretty easily, so kick it back to raw reads, and you can also go from raw reads to BAM by performing mapping. So those two processes are interchangeable. And to break it down, here's an example of one record in a BAM file. So it kind of looks overwhelming, but if you break it down piece by piece, it's not too bad. So we've got the read name. The next part of that is going to be that bitwise flag. This is basically telling me that this read is oriented on the opposite strand and that it was filtered out for some reason. Got the chromosome, the position on that chromosome, the mapping quality, so pretty low mapping quality for this one. This is that cigar, that representation of matches and mismatches. These next three have to deal with the next reads and template length and that sort of stuff, which aren't included in this particular BAM from 1,000 genomes. Then we've got our nucleotide sequence, and then here's that representation of our quality scores. And then at the end of the file, we might notice there's some additional fields that have been appended, some information about the sample read group and that sort of stuff. So there can be quite a few of these depending on what you're actually using. Okay, so we take our raw reads, come off the machine, and then we place it in a genomic context, and now we have everything we need to actually call our variation. Now, once everything's been aligned, we actually take a lot of this information to go through and pump it into a statistical framework, and we're really interested in things like, the mapping quality or some measure of confidence about our placement of the reads in a reference. If we're not really sure about that a given read should be in a location, we might therefore expect that information contained in the read to then have lower weight in our statistical analysis. Things like depth, the number of reads that exist at a given position, the number of alleles that exist, that sort of stuff. And you might be familiar with popular programs for variant calling. So the Genome Analysis Toolkit, GATK out of the Broad, FreeBayes, SAM tools, BCF tools, there are a number of these. And just to give a one-slide introduction to variant calling, imagine up here at the top, you have a reference sequence in blue, and then you have a bunch of reads that are mapped to that particular location below here represented in black. And what we're really interested in is asking the question whether or not this reference allele T is a T or is not a T or some other base in our sample that we see the reads below. So you'll notice that in many cases it looks like it's a G. So there are lots of Gs at this particular position, but then there's also some error associated with it. So some are Ts. There's a variety of reasons that we might expect to see this mismatch. Things in the laboratory can cause errors to be introduced. Things in actually generating the raw data itself can cause the errors to be introduced. Mapping can cause errors, all that type of stuff. So because we're interested in using statistics to do this, we can incorporate that variation and that error explicitly into our models and kind of get a better sense about whether a variant is a true variant or not. So imagine that our universe is kind of like this, and we're going to take depth and mapping quality, the actual bases or the alleles themselves, this error model I just kind of outlined. And in a Bayesian context, we can actually take prior information. I won't give an introduction to Bayesian statistics, but you might expect that maybe we're calling variants in a particular population where we know something about that, like the distribution of alleles is shifted for some region. So we can actually incorporate that into our models. You funnel this into a big statistical machinery, and then at the end of the day, we get this term. So the probability of our genotype given our data, where our data consists of all these pieces of information. It's kind of a super fast overview of the general approaches in statistical variant calling. So VCF files, as you might expect, do contain a lot of information at the per site and per sample level. So VCF files are broken down into basically 3 sections. The first is a header, just like in the SAM and BAM file. Second, kind of part of the header, but technically distinct, is a header line, which identifies what each column is. And the third are a series of lines that report various metrics at a particular genomic position, and then the actual genotype calls for the samples that you're working with. For example, the header looks something like this. So represented in this particular file, we've got info fields, which basically define what all variables are for information that might be reported aggregated across genomic position. Second is the format field, which defines the variables that are actually going to exist in individual sample level genotype calling. Third is the reference. This, in this example, contains a little bit of information about the reference genome that was used to call these variants. And then this very last line is that header line I was talking about, which defines what the columns are. So let's take a couple records and actually just break it down piece by piece. So this is the actual data that we're working with in a VCF file. So first we got the chromosome, so on chromosome 1. Then for each site that we're interested in, we've got the genomic position. Third field is an ID field. In this case, since it's from 1,000 Genomes projects, it's actually already been annotated with dbSNP IDs. You have the genomic reference allele, the alternative allele, so that's actually the variant that we're expecting at this position. This next field, which is represented as a dot here, is actually, it's what's called a quality field. And this is another Phred scale probability. Basically, before we even call a variant, we're interested in the likelihood of a variant actually existing in the first place. And so some of that information takes place here. So whether any filters are failed for this particular site. And then this is the aggregate information that I was talking about. This is aggregated across all individual samples. So in here we see examples like the number of total alleles, the depth at that position, et cetera. And it's separated with semicolons so we can easily parse it informatically. Format is going to be the structure of what the genotype calls for the individual samples actually look like. And then the final part is the genotype calls themselves and any associated information. So here we see that these samples, this particular sample at these positions, are all homozygous reference, and they have depth ranging from 6 to 0. Now, I should mention that we can apply filters after the fact that actually can clean up the data to some extent. So there are basically 2 ways to kind of bring everything home. To look at data in a VCF file where genotypes have been called. The first is by site. And in this context, information is aggregated across all samples, and all metrics are defined in that header in those info fields. You can really see what you're looking at. Now, filter applies to the site, not the genotype, although we can filter out particular genotypes that might be low quality for a variety of reasons. So population databases like Nomad, for example, might be distributed this way, where they don't include individual level identification, but it does include aggregate, say, allele frequencies across different subpopulations. Alternatively, we can look at it by sample. And by sample, basically, we're interested in the genotype of a particular sample. This is exactly what you would expect for, say, clinical purposes or research purposes, where we're really interested in the actual genotype calls of a, say, patient, for example. And then we can filter this using a variety of downstream efforts. So just to wrap everything up pretty quickly here, there are a few additional considerations. So a bioinformatician like myself, depending on what I want to do, might provide and execute a series of filters at a number of steps. So I might be removing low-quality reads, trimming low-quality bases, et cetera. And what this lets me do is really dial it in and come up with a set of variants. It's really rock-solid variation at the end of the day, which is really what we want in the first place. Sequencing depth for genotyping, basically, it's worth noting that it's easiest to call homozygous reference. It's more complicated to call homozygous alternative, and it's most complicated to call heterozygous genotypes. And that's because we basically need more information. So you could imagine a circumstance where you're trying to call a heterozygous site. In an ideal world, it's going to be 50% one allele, 50% another allele, but of course there's that statistical error, and that error on our estimate actually decreases as we increase the amount of data, that is the depth of that position. Here I'm talking about calling single nucleotide or indel variation, but we can also call haplotypes. And of course, haplotypes are more computationally intensive to call, but are really useful for downstream statistical analyses where we need phase alleles, phases implicitly as a result of this. And finally, we can also do joint genotyping. And you might have heard this before. This is basically taking multiple samples, genetic variation, and then incorporating it in to really dial in our genetic variation while using this additional information about allelic states across the population. Okay, so that's kind of a quick introduction, and I'll hand it back off to Kelly so we can get to Carla's part.
Great, thank you so much, Bryce. That was phenomenal. Appreciate it. So I'm going to, we'll hand it off to Carla for the second part of our talk today, and I will introduce her. So Carla Brigatti is a genetic counselor at the Clinic for Special Children, a nonprofit medical home for Amish and Mennonite children with genetic or complex medical disease. In addition to clinical counseling, Carla organizes and oversees the clinic's genomic diagnostic program, interfacing with families, clinicians, and scientists on genomic diagnostic testing and results. She performed her undergraduate work in biochemistry and biophysics at the University of Pittsburgh and received her master's in human genetics from Sarah Lawrence College. She's a board-certified genetic counselor and a member of the National Society of Genetic Counselors. Okay. Take it away, Carla.
All right, thank you so much, Kelly, and a big thanks to Kelly and the organizers for this talk, and Bryce for being sort of the back end of the house, if you will. I'm going to be the front end of the house if we're thinking of restaurants and food. And basically, I'm going to have the privilege today of talking to you guys a little bit about annotating and filtering variants. Just a little bit about me. Unfortunately, I don't have any conflicts of interest to declare. I'm still looking for those. And what I want to do today is sort of a fun exercise, really taking something, this is a very simplified version of a very complex exercise, and that is really sort of going through the tens of thousands of variants that can be generated in next generation sequencing and filtering those out and deciding or identifying the relevant subset of those variants to the disease of interest that you're studying. So really going from A to B. And you know, there are several software tools that can really use the VCF file as an input, and they compare that information against a large collection of annotation databases that will associate a variant with some type of function, a process, a pathway, or a disease itself. And so today what we're going to do is really sort of review some of the guidelines, the tools, and the resources that can identify variants with a potential association to the phenotype that you're studying. And I thought the best way to do this, what I find most helpful, is to really sort of do it through the lens of an actual case. And so what I want to do is we'll actually go through the process of next generation sequencing for a child we saw here in clinic, and the process of analysis and filtering the VCF variants into where we think we found the diagnosis for this child. In this case, it was a 6-year-old boy. He participated in in family-based whole exome sequencing. A lot of times, labs will do single or trio cases. When possible, we try to include unaffected first-degree relatives here. This child presented to us for several concerns. He had hydrocephalus. He had agenesis of the corpus callosum, was having some seizures. And in addition to that, he had some hearing loss, strabismus, and overall, not surprisingly, had significant global developmental delay. The family history itself was non-contributory, and at the time that they came to see us, he had one healthy sibling. So when we did the exome sequencing for the family, the VCF for this particular proband returned more than 8,700 variants. So obviously that's going to be way too many to sort of filter through one by one. So we used a host of different criteria to really prioritize those. The three that I'm going to talk about in a little bit of detail today are population frequency, functional impact prediction, which you often hear referred to as in silico tools or computer analytic tools that will do that for you, and inheritance models. And so this sort of metadata basically is an enhanced assessment of those variants that will tell you when they're likely to impact function. They'll know things like the known allele frequencies, how frequently they're involved in a human phenotype, and the predicted functional impact as well. So in looking at population frequency, in general, we tend to think that rare suggests that there's a functional effect, that there is a population or a selective pressure that non-functional or altered alleles or genes will have a deleterious effect in the sort of well-being of that person and reduce their reproductive fitness. The allele frequencies across populations have been identified in a number of different subgroups. These are just some of the more common ones that have been done. There are also sort of individual or smaller subpopulations that have been studied. Here at the Clinic for Special Children, since we study and work so closely with Amish and Mennonite individuals, we have our own sort of database of exome variants for those two populations that we frequently refer to when we're doing this type of work. So public databases that include this type of information you may be familiar with are those, for example, from the 1000 Genomes Project, ESP, which is the University of Washington effort, called ESP stands for Exome Sequencing Project. NOMAD is the latest and greatest kid on the block. That's over 120,000 different exome sequences that have been are available for those to review. And something to keep in mind is that when someone is called healthy, that's a presumption you have to make with a little bit of a grain of salt. And that is sometimes issues like incomplete penetrance, variable expressivity, and so forth can influence that. So someone may be called healthy, but in fact be a carrier for a condition that you're interested in. The functional impact prediction, this is our in silico tools. These sort of really will identify ways that, or predict ways that this variant is going to affect the canonical function of the gene, of the protein, and will often examine things like site conservation scores. And basically, these are mathematical or statistical computational tools that really study biological data. Now, there are all kinds of variant effect predictors tools that are constantly in use, and new ones that are being developed all the time. And two mainstays that you may have heard of are called SIFT and polyphen. And in general, just the thing about these is that they shouldn't be used in isolation, and unfortunately, it's not a perfect world. They can sometimes conflict with one another. Other common ones that are often used are other programs such as a CAD score, mutation taster, likelihood ratios, conservation prediction tools, and so forth. And there are really no standards for these filters, so some bioanalysts will prefer certain tools over the others and so forth, but basically, in general, majority rules in this type of case. So, considerations in these are basically what functional class the variant belongs to. Is it a truncating or splicing variant? Because that could potentially have a high cell impact. There are sometimes false positives due to sequencing error and selection constraints. And then question of how tolerant is this gene to loss of function, and how frequently is it conserved as such in other species as well. So this is just a picture of sort of the various tools. There are a number of different tools on the left-hand side, and a little bit about what those tools mean, what the letters mean on the right-hand side. And in between, you can see in this particular variant, all of the scores agreed that this is probably not one that would have an impact on the function of the gene and is likely not deleterious in nature. One thing to note about, if you look on the right-hand column, in the middle, you'll see there's a box that has N, and that's not an error in the software itself. The reason for that is that that is a prediction based on several isoforms of that gene or different transcripts of that gene itself. And so some predictive software will sort of examine all of the different transcripts and identify it for each one of those. So occasionally those may be conflicting within that region itself. This is just an example of one that is pretty uniformly pathogenic. You can see a lot of Ds for damaging, you can see Ps for pathogenic, and A's for automatic. The way that they're sort of assessed or assigned is a little bit variable depending on which tool you're utilizing. But you can see in this case, the scores came back pretty uniformly that this would be a deleterious variant. And you can see things like CAD score GURP, which looks at things like evolutionary conservancy, will have sort of scores that have a range, and the higher the range or the closer the range to the extremes, then the more you might say something is damaging or not damaging. And sometimes, as I mentioned, those scores can be conflicting, and in this case for this particular variant, we see some software, the tools that predicted, in silico tools that predicted it would be damaging, and others that did not. And so again, they're not used in isolation. In silico tools themselves alone should not be used to determine whether a variant is pathogenic or not, but can be an important piece of the puzzle in deciding that. One of the things to always think about is segregation, and that is if you have a variant that you're interested in, does it segregate appropriately within the family? And so working with that segregating variant frequency is another useful way that you can filter and reduce variants for analysis. So this is obviously only possible when you have other family members that you can compare to, parents and or other siblings or other affected family members. And of course it can throw you off because things like delayed onset, incomplete penetrance, variable expressivity of the phenotype, not phonotype, should always be taken into account when examining this type of information. But there are tools that really do perform filtering based on a pedigree and on a specified inheritance model. So if you're saying we expect this disease to be autosomal dominant, autosomal recessive, X-linked recessive, or what have you, based on the pedigree itself, then there are tools that can actually perform that filtering based on that information primarily. So if you have a number of variants that you can sort of minimize them, we want to know how do you prioritize those? How do you decide which should be at the top of your list for consideration? And so, important pieces of information to consider are how they're implicated in disease or in a related gene. So important tools to go to for this are databases and other publications and so forth. So the ClinVar database, of course, is one that will tell you whether this variant has been assigned a pathogenicity score or not, if it's known. The Human Genome Mutation Database is a sort of ongoing catalog of those variants as well as they've been published or reported as pathogenic or benign. PubMed is always a great source for examining the latest in medical literature to find out if there is a publication or a paper that might shed some light on a particular gene or a variant within that gene. OMIM is tried and true in terms of looking up conditions that may be associated with a human phenotype. Dysgenet is another resource in that capacity. And so, assessing these candidate genes in the context of existing biomedical knowledge and known pathophysiology is a really important step in making that list manageable in doing so. And if you're interested in a gene but it's not found, there's not a lot of information in these resources that I mentioned here, then you can always think, well, is it expressed in a tissue of interest? Does it interact with a protein that we know is associated with a specific disease that we're studying? And so these are questions that you can go through and sort of really try to identify that small list of variants that you want to be interested in further study. So if I go back to that case that I mentioned in the beginning, and to this sort of generate from those 8,700 variants that were identified in the VCF, narrowing it down to a number that this is sort of the shorter list that we came up with. There were a number of genes that were inherited in a de novo fashion, and there were those that were found in a homozygous recessive fashion, or that were in compound heterozygous state. And so you can see here we took into consideration what the actual frequency of this variant would be, in these variants would be in a number of different population databases. We checked to see how conserved it was with other species and so forth, and then what the prediction would be based looking at those number of different in silico tools that I had mentioned a little bit earlier. In doing so, what we were able to do is sort of narrow this down a little bit. So, oops, sorry. We went to, if you look at that first variant that was on the list I mentioned, this is a variant in a gene called ZNF462. It's a zinc finger family of proteins. It was de novo. This variant was not identified in either parent. It was not found in any of those common population databases, nor within our own internal database. It was predicted to be conserved, so it's probably a gene that's not that tolerant at this position to change. And those in silico prediction tools had predicted this would be deleterious. We knew this gene belonged to a zinc finger family of proteins. It was involved in transcriptional regulation, which is obviously important. And we knew that it had been expressed, you can't see it down below, but it actually is expressed in a number of different tissues that would be involved with this particular disease pathway for this child that we were studying. And so, in fact, when you go to the literature in PubMed, and this is a very recent publication, you find that, in fact, ZNF462, or haploinsufficiency of that gene, is associated with a complex condition of craniofacial anomalies, abnormalities in brain development, developmental delay, and ptosis. And so we really felt like, you know, this particular variant of interest, while it was not listed in any of the databases of variants associated with disease, in ClinVar or in the Human Genome Mutation Database, this piece of information in the literature was actually very helpful in going back and seeing, is this a really good possibility for our patient? We did go through and just ensure, this is just a picture of the BAM file. You can see that the father, the mother, and the sister don't have any alternate reads at that particular position, but the proband had a pretty good read depth and high quality calls at that position. So we felt like this was a legitimate call on the part of the testing itself. And so ultimately, based on segregation analysis, our in silico tools that we used, population frequency, we were really in that paper, we were able to predict that this ZNF462 variant is likely pathogenic. So ultimately what we did, standard practice, is we Sanger sequenced it, confirmed that it was in fact a real call. We called the patient back, rephenotyped him, and really demonstrated or found that this was a very good genotype and phenotype match. So ultimately we felt very satisfied that while this was not a gene that has a lot known about it yet, or has been found in high numbers in the patient population and so forth, that this was a pretty good candidate and likely the underlying cause for this child's complex phenotype. So, you know, just closing thoughts here is that we really gained a lot of insight in how to interrogate this genomic information. And ultimately, there are a lot of tools and a lot of domains that we can utilize that can give us evidence to implicate a variant in this type of a case. There are a lot of limitations individually, but ultimately, the sum of them, or the aggregate of them, they really do act like a funnel, and they distill those thousands, tens of thousands of variants into something that's clinically meaningful. And so ultimately, utilizing these tools is a very big help in narrowing the focus to those variants that are relevant to this case in particular. So those are the things that I wanted to talk about, and I thank you for your time and attention.
Great, thank you so much, Carla. That was really fantastic. I'm going to go ahead and share my screen again just to remind everybody of how to ask questions. So over here, so on your control panel that shows up usually on the right side of your screen, there's a box that says questions. Sometimes you have to actually expand it, but you can enter any questions there, and we will take question and answer for the next 10 minutes. I have a couple that I'll get started with. First is, there's a comment, thank you, Bryce and Carla, for all this great information. I can imagine that I will love to use this information for reference in the future. Will the slides be available to use in the future? So I can answer that one. Yes, we will provide a copy, a PDF version of the slides. If anybody is interested in receiving those, please reach out to me directly. I'm Kelly Hagman at Ambry Genetics, and my email, I should have put it up on the screen, but it's actually kfarwell, K-F as in Frank, A-R-W-E-L-L @ambrygen.com. And next question is, there's a question, what program or resource do you use to review if proteins are expressed in particular tissues? So Bryce or Carla, do you guys have any suggestions for that?
So there are sort of, I know, different ways that you can see if it's expressed in a tissue or organ of interest. There's something called the Gene Expression Omnibus Profile that can be generated. There is something called a GTEX, GTE in capital letters, small x database that can have that. And then there are expression atlases as well that can sort of give that information. There's other databases that I occasionally go to, things called like Malacards and so forth, that will tell you, or Gene Ontology Enricher, that can tell you sometimes whether certain organs or tissues of interest would express that particular gene. Bryce, I don't know if you have any other thoughts on that.
Yeah, those are exactly the ones that I was going to mention. I guess I'll just add that there are a number of public resources, especially if you're in academia, like gene cards, for example, that can go through and not only includes expression information, but also a ton of additional information aggregated from different databases, methylation patterns, different tissue-based expression, so general information on the gene, all that kind of stuff. So generally, quick Google searches can bring up some of these big aggregate websites that basically take all this information, so you don't even need to get into the nuances of it as well. But the ones Carla recommended are exactly what I would have suggested.
Okay, we have another question. What do you recommend for publishing your case to add to the general public knowledge?
Well, that's a great question. I'll just add my 2 little cents first, and that is one of the quick ways to sort of make that public knowledge out there, to share this information, data sharing. That's such a question, very timely one, is to put it on ClinVar, put it on those databases where they're aggregating that information and collecting it. And those databases are parsed out in a way that you can, describe the specific case, why you have decided that this particular variant is pathogenic or not, and sort of levels of evidence that supports that call one way or the other. So that's a quick and easy way to do it, and it's one that I know I utilize all the time. I'm always going to ClinVar to see, has this been reported elsewhere, and if so, how has it been described, and so forth. That's one. Publication is always an important step. Getting that paper published is a time-consuming process, but an important one for really disseminating this type of information. I mean, in the case that I talked about a little bit earlier, that paper was just published in August of 2017. So if this had been a case that we had analyzed some time ago or seen some time ago, we wouldn't have had that additional piece of information to utilize in really saying with more confidence that we felt like this is the underlying variant causing the disease. And so, I can't emphasize enough, it's a process, but it's a really, really worthwhile one, I think, one that we all benefit from.
Great, thank you. Okay, we have another question. I believe this one's for Carla. So thanks for a great presentation. Do you think the testing lab should do this variant analysis, or do you expect that you'll do your own analysis after getting results from the NGS lab?
Great question. So obviously, if you're sending this if you're doing testing with a laboratory, then it's really on them to be able to provide the highest quality information. And that's obviously somewhat subjective in some cases in terms of interpretation of those variants. But I think where it falls on clinical genetic counselors is just a little bit of savvy in terms of how that process is happening and how those decisions are made. Sometimes it'll be, you'll receive a report with a variant of uncertain significance, and it's important. It's like, well, why did this not meet, their criteria, internal criteria for calling it pathogenic? Or is there additional information that could help the laboratory identify that? So I think it's really sort of a partnership. One is, I think, the filtering and that list that's generated in the analysis process really should be the laboratory that does that. But I think having a lot of transparency in that process and looking through that, or having an understanding of how that information is generated, is really helpful for the clinical counselor, I think, in terms of explaining those results to the family and the implications of that information, but also in generating the best result possible.
Okay, perfect. Thank you. I'm just going to add, if you have any other questions, please feel free to go ahead and ask them in the panel. But in the last few minutes, I'm just going to add a quick slide to add my information in case anybody has any further questions that they think about. So I'll put my email address along with the Genomic Technologies email address. Okay, so those are the email addresses. If anybody would like to take them down, you can feel free to reach out to the Genomic Technologies SIG listserv or directly to me. That's my address there. Again, thank you so much to both Bryce and Carla for a fantastic talk. Kim and I, as co-chairs of the Genomic Technologies SIG, did do a survey a couple years ago, surveying the general genetic counseling listserv and all the certified genetic counselors to find out what needs would be most needed in terms of genomic technologies education. Bioinformatics listed right up there at the top, which I think explains why I'm getting such good responses already popping in my email box about asking for slides and for more information about this. So again, we really appreciate it. It's much needed, and if you have any further questions, anyone, please feel free to reach out to me directly. All right, thank you very much and have a great day, everyone.